I think we'll make a start. We still have a few participants just joining us, but we'll make a slow start so that those who are arriving can settle in. Welcome, everybody. Thank you very much for joining the ME Genetics Centre of Excellence webinar. For those that don't know me, my name is Sonia Chaudhry. I'm Chief Executive of Action for ME. I'm a member of the Centre of Excellence Partnership Board and also involved in a number of different projects, including Decode ME and LOCOME. I'm really pleased to be back here again with you since our last webinar in October. And today we're going to share some of our latest updates from research linked to the Genetic Centre of Excellence. By way of reminder, the centre is a virtual centre established to accelerate genetics research. And to find out more, you can go back to the webinar that we held in October. And at the end of the webinar, we're really excited to be able to share with you a link to our new website. We're launching it today. There's more work to be done. So we're going to continue working on that and building it up over the forthcoming months. As you may already have seen, the webinar is going to be recorded. You won't be seen, those of you that are here listening and joining us today, but we do want to make this available for people afterwards. And we know that some people may stay for a little bit, dip in and dip out. So that's absolutely fine. The recording will be available on the Genetic Centre of Excellence website as soon as we can get that ready for you next week. We will have some time at the end to answer questions, and thank you to those of you that sent questions in advance. If you do want to ask a question, you can pop those in to the chat. I'll be monitoring those with my team, Daphne, who's our research manager, who's on the screen at the moment, and also Alice, who is working with us in the marketing and comms for the Decode ME study. So they will be disappearing shortly, but they're working really hard in the background, helping make sure that the IT and me work smoothly as possible and that we pick up as many questions as possible. So I'm really pleased to be able to introduce Dr. Audrey Ryback. Audrey's a research fellow working under Professor Chris Ponting at Edinburgh University, and I'm doubly pleased because Audrey was our first Claire Francis Research postdoc fellow and has been working on a range of different things. So it's really I'm really delighted to be able to introduce Audrey today. So I'm just going to hand over to you, Audrey.
Thank you for the introduction, Sonia. Let me just share my screen. And can everyone see my screen and my cursor? Yes, okay, excellent. Yeah, thank you so much for inviting me to present today. Today I'm talking about an update on a project that I presented at the last webinar, asking the question, do ME blood factors change the behavior of cells? Now, as Sonia already mentioned, I'm the first Claire Francis Research Fellow funded by Action for ME. It's a real honor to hold that title. And the overall hypothesis of my fellowship has been looking at whether ME blood factors change the behavior of cells. I became interested in this question because for some time there's been a hypothesis in the ME research community that there is some sort of effect of ME CFS plasma or serum on cells when they're exposed to serum from patients in the lab compared to serum or plasma from controls. And this is really nicely summarized in a blog post by patient advocate and science communicator Simon McGrath, which you'll notice is from 2019. So this hypothesis has been around for quite some time. And a range of small studies pointed towards this kind of effect, but I was particularly interested in the question of whether healthy cells change their mitochondrial function exposed to ME serum when exposed to ME serum compared to healthy control serum, because the studies that suggested this I found particularly compelling. So I'm just going to introduce how we test for mitochondrial function, so that you can, to give some context for the experiments I'll describe later. So in the process of generating energy in the form of ATP in the cell, the mitochondria consume oxygen in this process. So I've highlighted the oxygen here in red when they produce energy by aerobic respiration. Now, we can test mitochondrial function by interfering with this process and adding different toxins that disrupt different parts of the mitochondrial energy production machinery. And you get a corresponding change in the oxygen levels in the cells. And this can be measured on a machine called a seahorse bioanalyzer, and you get this characteristic curve where it measures oxygen consumption rate along the y-axis, and over time, as these different toxins are added, you get either decreases or increases in oxygen consumption in the cells, and this corresponds to different aspects of mitochondrial energy production. So the first aim of my fellowship was to replicate a finding published by Flug et al. Back in 2016, where they saw increased maximal respiratory capacity in muscle cells that had been exposed to ME serum compared to healthy control serum. Now they performed, what they did was to expose healthy muscle cells to ME or a healthy control serum, and then perform these mitochondrial stress tests. And they saw differences actually across the different conditions that they measured. But in particular, I was interested in their finding under conditions of maximal respiratory capacity, because the magnitude of the difference was very, very large. So one caveat here is that the sample size was small. So they had 12 healthy participants and 12 people with ME. And so I set out to replicate this finding because replication studies are really essential to strengthen ME research. So many ME research studies have small sample sizes, and this is often due to a lack of funding. But even if the studies can be carried out very, very well and the science done very well. However, studies with small sample sizes are less able to discover true differences and more likely to show differences by chance. And they're also less likely to reflect results from the broader population. So it's more difficult to generalize results from small studies. So therefore, performing a successful replication would give us the confidence that the result is generalizable and is telling us something about the biology of ME. So in order to make our replication result as definitive as possible, we made sure that we had good statistical power, meaning we had a large sample size. So we had 67 people with ME, 53 healthy controls, all female. We performed 15 technical replicates per participant and generated over 1,900 of these seahorse mitochondrial stress test traces. All of the work was performed blinded, and so we didn't know who was a case and who was a control when we were performing carrying out the experiments in the lab. And we pre-registered our study on the Open Science Foundation, meaning that we declared in advance how we were going to carry out the study and what we would consider to be a positive result. So the graph on the right here shows one of the examples of the plates that we ran. And here you'll hopefully recognize the shape of this curve again, with oxygen consumption rate along the y-axis and the changes in oxygen consumption rate as expected following the addition of these different toxins. Now, these are measurements from individual wells. They're colored by the participant whose serum was that these wells were treated with, and yes, and this is to show, basically to illustrate that the assay was working as we expected. And you'll notice these are blinded IDs, so as we were carrying it out, we didn't know who was a case, who was a control. When we came to unblind, we found that there were no significant differences between these healthy muscle cells that have been exposed to ME serum or healthy control serum. This was our primary outcome of interest. So the oxygen consumption rate under conditions of maximal respiratory capacity. And these are the averaged values per replicate here on the left for healthy controls, on the right for our participants with ME. And you can see by eye already that these two distributions overlap quite a lot. And indeed, when we test them statistically, we have a p-value of 0.2. It is not statistically significant. This is the same result, but this time we've accounted for sources of technical variation in the experiment. And again, there is no difference between the two groups. Another way of showing this result is to look at each participant individually, and this is what this graph here shows. So each box corresponds to one participant, and they are colored in blue, the healthy controls, in red, the participants with ME, and grouped by sample set. And the point that I'd like to make here is that you can see there are differences between individuals, but there are no global or systematic differences between the people with ME and the healthy controls. So what does this result mean? It means that it is unlikely that the original finding of an effect of ME serum on muscle cell mitochondrial function reported by Fleury et al. Is true for ME in general. Now, I just want to be very clear that I believe ME/CFS is an organic illness, and there are biological processes involved in this illness, and that this result does not change that fact. I understand that the instinct might be to be disappointed by a negative replication, but it is actually a really important result because it tells us that this is not the place to be looking. And so we need to look at the problem differently. I also wanted to say that we can't tell from this whether mitochondria are affected in people with ME, because what happens to the muscle cells in our experiment doesn't translate one-to-one to what happens in people's bodies. And based on our result, we think that the hunt for ME disease factors and processes should be done using methods different to the ones that we've just presented. So one of these methods that we're now pursuing is, once again, a replication study, this time hunting for biomarkers using proteomics. So together with Dr. Awak Hamza and Shad Benches, we are using O-link proteomics and a few other assays to replicate a set of ME blood markers that they've identified in the UK Biobank in people with ME. This has been published as a preprint last year. So we're looking to validate these markers in the independent cohort that I have plasma samples for, and that's sort of the primary aim. And then following on from that, we will explore whether this set of biomarkers might make for a predictive panel that could be used as a biomarker panel for ME. So the take-home message here is that we don't believe that ME serum, when applied to healthy muscle cells in the lab, affects mitochondrial function, and that the replication studies with large sample sizes are essential to establish research findings in ME that are generalizable and therefore tell us something about the underlying biology of ME. And with that, I just want to finish and acknowledge everyone who's been involved in this work. It's been a big team effort, and the results that I've presented today have been submitted to bioRxiv. Unfortunately, the paper is not live yet. I was hoping it would be before the webinar, but it should be live really any minute, and you'll be able to look at the results in more detail there. I'll be posting that study and a lay summary of the study on my Blue Sky account, so feel free to follow me there if you want to stay updated with that. And thank you.
Thank you very much, Audrey. Always very interesting, and you stuck perfectly to time as well. There are a few questions coming in. We will be able to answer questions that relate specifically to the presentations that you're hearing today or the work of the Genetics Centre of Excellence. So apologies if you have questions that aren't related to that, but you can always go to Action for ME Information Support Services and our website and some of the other charity websites, including the 25% ME group, if you want more information. Audrey, I know we've got some time a bit later for questions, but one question I think it'd be great for you to answer immediately that's come in is, what is in ME serum?
Oh, apologies for not explaining that. That it is the liquid portion of the blood. So if you allow whole blood to clot, and you spin it down, the liquid that you collect from the top of that is the serum. So it doesn't contain any cells, but it contains lots of other proteins and signaling molecules and metabolites.
Fantastic. Thanks so much for sharing that, Audrey. And while Audrey's stopping sharing her slides, I'd now like to introduce some additional guests that we have. So Steve Gardner, Jason Sardell and Christina Taylor from our partners at PrecisionLife. I'll hand over to the team and I'm sure they'll introduce themselves as they work into their presentation. Thanks.
Steve, we're struggling to hear you here.
Aha, that might be better. Apologies. Thank you, Sonia. Really appreciate the opportunity to present some of this work. Some of you may have seen this at the recent IMEC conference, but we wanted to take you through some of the work that is going on the back of the Decode ME study and various other collections. I'm joined by two of my colleagues, Christina Taylor, who is primarily responsible for many of the disease studies and interpreting the results, and Jason, who leads on quite a lot of the analysis of data and the development of new methods. They're going to be contributing through the presentation. So I'm going to get started, and what we're going to talk about are a little bit of an introduction to a project called LOCOME. This is a project that is in partnership with Action for ME and Decode ME and the University of Edinburgh, Chris Ponting's group. We're going to show you some of the results and tell you how analyzing these data helps us understand the biology of the disease and hopefully find new diagnostic and drug solutions. We're then going to try to talk through what this might mean for ME and long COVID patients and finish up with a little bit about what we're going to be doing next. There is a web page with all of the information that I'm going to be sharing today. You can reach it through this link, or we'll give the information to Sonia afterwards. And I just want to emphasize that this is a village. This is a very collaborative effort with a lot of different players in the mix. So Christina, do you want to take us through the basic methodology and some of the initial starting results?
Sure. Yeah, so this all began back in 2022 when we performed a genetic analysis of an ME/CFS population using UK Biobank data. Throughout this presentation, we might use the word SNP or disease signature. What we mean here is that we are looking for mutations in genes, in DNA, that is particularly associated with disease. So the key questions we're asking here are, what mutations are associated with ME/CFS, and can we group patients by common mutations in order to understand more about their specific biological mechanisms that might impact their own disease etiology and their symptoms. So this MECFS case population was defined using the results from the pain questionnaire in UK Biobank, specifically including those who had answered yes to having experienced post-exertional malaise, or PEM. We then need to create a control population, so a group of people who we use as a comparison to understand what the genetic differences are between those that do have MECFS and those that do not. These controls were sex and genetic ancestry matched against the case population, meaning that if our MECFS case group is 70% female, so are our controls. And these controls also had no evidence of chronic fatigue, pain, or sleep-related disorders in their health records or self-reported data. And what we did is we took that genetic information and we performed something called a combinatorial analysis of it. And what that has shown here in the center of the screen is a disease architecture that was generated by clustering the genes or the mutations on the basis of the patients which they co-occur, i.e. these are genetic subtypes of MECFS. And we find 15 distinct subgroups here, each denoted by a different color. And within these 15 subgroups, we find 14 unique genes. And what we can do now is show how each subgroup or explore how each subgroup, their biological mechanisms differ and how those differences might relate to their unique symptoms experienced. And before we dive into each group, I just want to highlight the importance of collecting this large-scale data for chronic complex diseases like MECFS. We were able to do this because of resources like UK Biobank, which has been a great starting point for our work in MECFS. But the data collected there was not specific for MECFS. It didn't contain the right questions, the right clinical data. And that really highlights the benefits of things like the DeCODE-ME work for any future research and study into MECFS that we're currently doing. So I'll pull out a few examples from these groups here, and this has all been written in much more detail in our publication that's highlighted at the bottom of the screen. This green group here represents a genetic cluster that represents just over a quarter of that UK Biobank MECFS case population. And the critical gene mutation at the heart of this cluster of variants here is found in the gene called ACAP1. Now, ACAP1 is a regulator of mitochondrial energy production and release, and mice that don't have this gene or it's deleted in them are unable to meet the increased energy demands brought on by exercise, so they experience PEM. And as expected, when we look into the clinical data of these patients that have this genetic background and the responses that they gave as part of the various different questionnaires, we can see that they are much more likely to present with that classic kind of fatigue PEM phenotype when we compare their answers against other MECFS individuals that are found in different genetic subgroups. So I think we've got a few more examples coming up here on the screen. Blue group here on the left-hand side represents a cluster of genes that are found in a smaller proportion of patients with MECFS. And we're focusing in on one of these genes called GAP3, which is a neurotransmitter transporter. And again, there's biological evidence in the literature that dysregulation of this transporter results in cognitive impairments, in stress vulnerability in mice. And again, we see evidence of that in some of the individuals' self-reported data in the UK Biobank who belong to this subgroup. As well as a significant increase in their phenylalanine levels, which is a precursor to many neurotransmitters that's evident in their metabolomics. And then, as Steve's put on the slide here, we've got several other examples from different genetic subgroups. Again, there's more time and more detail included in our publication here, but we see key drivers or signals around things like the insulin receptor, which is represented by the pink group, in the clock gene involved in sleep dysregulation and a possible additional fibromyalgia co-association. And then in the red group here around a tachykinin receptor called TACR1 that is much more involved in POTS and dysautonomia and also found in our long COVID study.
So this is fantastic. It's starting to allow us to understand a lot of the drivers of potential drivers of disease biology. So we did that. PrecisionLife did that study, and it became a reason and an evidence set that we could use to support a grant application that we put into Innovate UK. We were lucky enough to win that, and it has funded 2 years of research. We are now about 18 months through that project into replicating, as Audrey said, the critical importance is to replicate these findings, replicating the results in different patient populations, particularly where they have more diverse ancestries. And that really is where Decode ME becomes incredibly useful. As Christina said, there is much deeper survey information associated with that data set. Unfortunately, we weren't able to pick up long COVID patients from Decode ME, but as we'll show you, we have done that in other areas. The whole intention of the project is to develop more insight into the disease biology and prove that it is predictive. So we want to be able to develop a rapid, accurate, and consistent diagnosis, and also potentially to identify precision drug repurposing candidates. So in other words, have an evidence-led view that drugs that work in another disease area could be useful to certain patient populations within the ME community. And this is massively important. As everybody on this call knows, these are hugely debilitating diseases that affect hundreds of millions of patients around the world. And we don't have any approved diagnostics, and we don't have any approved what we call disease modifying therapeutics, things that treat the causes of disease rather than their symptoms. This is going to change. This is a slightly old slide, and I think Chris and the Decode group will hopefully change the metrics here. But other groups so far have not managed to replicate any of the results from genetic analysis. We've been really successful on this, and I say that in the light of some evidence that I'm going to share later. But we found 14 genes, as Christina said, in that first study, and 73 genes associated with long COVID. What is really interesting is that we've shown that 9 of these ME genes also show up in the long COVID population. So 9 out of 14 show up in common. And we have every reason to believe that this may even go up as we analyze additional populations. So this is really interesting and important work. It underpins genetically a suspicion that we're seeing from broadly across the community that there is a strong relationship between these two diseases. This is, this work came from a UK patient population in long COVID. We've published on this as well. I'll give you all the references at the end. The other thing that I wanted to mention was that we have found 13 drug targets where we have existing medicines. So we've got actually quite a number of opportunities to repurpose medicines in a very directed fashion, both for long COVID and ME/CFS. In fact, many of these repurposing opportunities would likely be effective in both diseases. Audrey again and Chris always mention replication of results. And actually, more than that, it's important that we see that results that work in one population actually work in populations with different ancestries or different socioeconomic backgrounds, different comorbidities. So our first job, and something that Jason's been spending a lot of time on, is making sure that these results reproduce. In long COVID, the good news is that over 90% of the genes that we found in the UK population that we were able to test in a US population, the All of Us population, reproduced. They had a positive association with the disease in that second population. That includes 7 of the 9 ME genes. We weren't able to test 2 of them. So if you remember, 9 out of the 14 genes that we found originally um, were present both in ME and in long COVID. 7 of those also reproduced, and we're still trying to test the other 2. And again, encouragingly, 11 out of those 13 drug repurposing candidate genes also reproduced between these data sets. So that's really fantastic. We're hoping to do many more of these studies. We're very close to being able to report on the results in ME between the UK Biobank data and the Decode ME data. We've got some technical challenges there, but we're working with Edinburgh to overcome those. What is also notable is, this may seem like a bad result, that when we move to Black and African American, Hispanic or Latino populations, we have a lower level of reproduction. Obviously, we would like that to be equivalent across all ancestries. However, that still is a much higher reproducibility number than you would get using a traditional genetic analysis. So this is really encouraging that we will be able to find mechanisms that could potentially be the targets of drugs that will benefit as wide a population as possible. Okay, so how do we go from that to diagnostics and drugs? Well, one of the first things that we need to understand is we found 200 mutations and 14 genes. How predictive are they? When you come to a new patient, if you see those same signals, does it mean that they're likely to have ME? So we basically take that data. We're actually taking 2 sets of data. We're taking the disease risk genes that we can see, and also we run analyses on the protective effects. There are genes that act positively to resist the onset of disease, and that obviously subtracts from your risk of getting a particular disease. We put the 2 together and we count how many times we see those risk signatures in patients on the left and controls on the right. And the bigger the difference between these, the more predictive these signatures are. The really good news is that our odds ratios are pretty high, unexpectedly high, given that there's no previous genetic associations. And this is great news, because what it means is we can go and test. Sonia, you have a question?
Yeah, I was just wondering, Stephen, sorry to interrupt and not wait till the end, but we've had a couple of questions around what are odds ratios. So maybe you could just explain that while you're on this slide for us. Thank you.
Jason, what's an odds ratio?
Yeah, it's sort of linked to probability. I mean, odds and probability aren't the same thing, but you hear a lot about odds in gambling. So if you have an odds of 2, that means it's twice as likely to happen. So an odds ratio is just a ratio of odds. If you have an odds ratio of 2, it's the odds of having disease if you're a case. I mean, the odds if you have it is twice as high as the odds if you don't have it, essentially. So bigger is better, and 1 means they're the same. There's no effect.
Yeah. So basically, if you have these risk signatures at this kind of level, broadly speaking, you're 4 times more likely to actually have ME, is the broad answer. But the great news is this is running on the type of test that was actually used in the Decode ME, i.e. you can do a cheek swab, a buccal swab. We can run it on a fairly low-cost genotyping array. We can look for these signatures, risk and protective. We can, for any individual, we can subtract one from the other, and it will give us an overall likelihood that they have that disease. Of course, what it will also do is tell us which of these mechanisms is contributing to disease inside that patient, and that will guide us in ultimately when there are drug options. It will drive us to which drug options are most likely to be relevant for that patient. So we're validating these at the moment. And Christina, do you want to take us through the clinical trial?
So we now have a collaboration with the Mitochondrial Institute, which is a research center of excellence based out of Salt Lake City in Utah in the United States. And we formed a very unique partnership with them in order to increase or jumpstart diagnosis and treatment options for patients with chronic diseases that are historically hard to diagnose and treat. They have a biobanking project called METX, where they are collecting data for these patients with chronic diseases like ME/CFS in order to create the world's largest data resource for patients with those diseases and clinicians and researchers studying them. From this, we are working with Metrodora on the MELO study, where we are currently actively recruiting up to 1,000 patients in the United States with clinically diagnosed ME/CFS or long COVID in order to evaluate the predictive accuracy of our test in identifying ME/CFS and long COVID cases. What we've shown here is we've pulled up a random selection of results from ME/CFS patients in the METX study that we have ran our test on. As you can see from the radar plots that are going to come up on the screen, there we are. Whilst each individual has the same clinical diagnosis of ME/CFS, the underlying mechanisms that we see that are most active in each person are completely different. And so we can see why there's such a variety in experiences reported or experienced by those that have ME/CFS. As some examples to pull out here, patient 2, their genetic background reflects genes and mechanisms that are much more likely to be involved in neurotransmitter dysregulation and fatigue based pathways. Whereas patient 3's plot, just next door, indicates a much stronger metabolic and inflammation autoimmune component. And this provides a completely new perspective for the clinician and the patient when approaching their form of ME/CFS. These plots or these insights into their underlying disease mechanisms might also go a way to explain their individual symptoms and what interventions might be better suited for them.
Thank you. That's really only half the story, though. So this is exciting. Obviously, we are very strongly of the belief that the community will benefit from having a definitive biological basis for the disease and having a test that would indicate to clinicians a very strong probability that that's what's causing the symptoms. But there is another piece, as we've described, with the protective biology. You know, we're not just all about those risk factors. Jason's been very active and published on this in the ME/CFS space.
Yeah, so it's one of the things we're really excited about at PrecisionLife is what we call actively protected biology. It's something we're really pioneering, and the idea behind it is as Steve was sort of alluding to, it's that many controls, many healthy people do have risk factors. It doesn't, and no risk factor in a complex chronic disease is 100% predictive. And when we look at patients who have many of these risk factors, there will be some of those will never develop the disease. So this opens up an opportunity to identify what we call like protective factors. So these are, in our aspect, in our studies, we're looking for genetic mutations that are inferring some resistance. Some they can't partially or wholly mitigate the effects of these genetic risk factors. Now, you could do that by just simply looking for, instead of as we do, looking for features that are enriched in the patients, look for features that are enriched in the controls. But the problem with that, of course, is that many of those controls are simply controls because they don't have these risk factors. So what we do in these sort of analyses is sort of the plot on the upper right that Steve put up and showed before. If we take our control group, we can identify a subset of these controls who do have many of these causative risk signatures. And then we focus our analysis just on those patients as well as those that protected cohort, as well as the ME/CFS patients who have similar numbers of combinatorial risk signatures. And using this analysis, we hope to provide some insight into protective biology, which can then be translated into potentially new prophylactic drugs, new mRNA vaccines, et cetera. Just novel therapies that replicate the same biology that these protective risk factors are inferring into the patients who have them. So we've done this for ME/CFS on our original UK Biobank cohort, published it earlier this year, and Christina can talk a little bit about those results.
Yeah, I think we might skip this in the interest of time, unless anyone's particularly keen. There is a whole paper describing these, and we have the literature in the back for them. But one thing to note on this is that actively protective targets may very well help everybody, whereas traditional drugs just fix something that's gone wrong in a subset of patients. These may actually end up helping most people, and that's a that's a really interesting kind of opportunity for us. I'm going to touch very quickly. I'm conscious of time. I'm going to touch very quickly on repurposing, and if you put all of the things that we've just described together, one of the things that we have an opportunity to do coming out of the trials for the diagnostic is to find patients who have a particular mechanism that seems to be going wrong, a particular signature. And given that we've already analyzed all of the drug pipelines of major biotech and pharma companies, and we know which drugs are active against those targets, it gives us the opportunity to recruit small cohorts of patients where we can trial these medicines. Now, we're focusing on generic compounds, things that have been on the market for ages and come off patent, so are relatively cheap, and they're small molecules. They're things that you can give as pills rather than injections. So they're very easy to dose, they're very safe, they're very well tolerated. And what we're really looking for is a response that says, yes, this therapy is impacting the disease in a meaningful way, not just a symptom, but actually the cause of the disease. And if we can do that, if we prove that molecules targeting this gene are going to have a beneficial effect, and that we can choose the patients who we can select, we can find, it's a better word, we can find the patients for whom this drug will be beneficial, that is a huge advantage for a pharma company coming into this space and bringing all of the machinery that goes alongside that, the attention, the effort, the money, to bring new therapies to market. So this is a really pragmatic, a very fast way of getting new drugs into circulation. We have many of them. We have many candidates, sorry. As you can see, many of them overlap both long COVID and ME/CFS, as I said at the beginning. Don't worry too much about these prevalences. These are minimum numbers. You can see that some of them will work in at least a third of patients. That's actually a pretty good result, but we expect that number to go up fairly significantly. But it does prioritize two points. Number one, the first drug that comes out in this space is very unlikely to work in 100% of patients. Symptoms in patients are so different, that's very unlikely. And second is, we have more repurposing candidates than we have clinical trial sites. And one of the things that we're all working on is trying to improve the scalability with which we can test these compounds. Okay, so to sort of finish off, what's happening in the next 12 to 18 months? Jason, do you want to pick up on reproducibility?
Yeah, I mean, one of our biggest focuses in the near term is going to be replicating our results. As Steve mentioned, we've done that in long COVID. We've published our replication. We want to do that in ME/CFS, not only in our original publications, but we have access to the Decode ME patient cohort. We've analyzed that through a combinatorial pipeline. And now we want to replicate it in other batches of Decode ME patients as well, and then prepare that for publication this year. And of course, we're most interested on these genes and these drug repurposing candidates and the replication across both long COVID and ME/CFS, but also a replication of any new novel drug targets that come out of this Decode ME analysis.
Yeah, and we're very actively working to bring these to do the clinical validation of these diagnostic tests, or we're calling them mechanistic tests. That will be in the US first because of the regulatory environment, and we will also be running clinical trials of precision repurposing candidates in a variety of places, hopefully including Australia, strangely enough, and some of the actively protective candidates as well. So with that, we'll finish. This is your beautiful team who have done all of this work, and there's a couple of rather fabulous collaborators that we couldn't not mention in the context here. And if anybody wants to delve into the science in a lot more detail, there are a whole variety of resources available where we've published this.
Great. Thank you, PrecisionLife team. We've got lots of questions coming in, so I'm going to fire out as many as I possibly can to the speakers that you've already heard, but also I'd like to introduce Helen Baxter from the 25% ME group, who also sits on the Genetic Centre of Excellence and has been involved in other things like Decode ME too. So before I come to a question for you, Helen, I'm just going to pick up a couple to the PrecisionLife team, just because that follows on. As you can imagine, there's a lot of excitement around the fact that you found potential drugs that could be repurposed. We've got questions around which drugs you found, how many of the drug repurposing genes were in the ME CFS gene set. Can you just say a little bit more around that and why those results aren't quite ready for people?
Yeah, absolutely. So we showed 5 candidate genes that we have prioritized, where there's a good overlap in 4 of them between ME and long COVID, and those are ones that we really want to chase down. We haven't, because of the IP situations around some of these, we haven't published on this work yet. We are very actively in discussions with a whole variety of different centres to actually go and get these tested, and they will come out in due course. Christina, I don't know if you want to talk about any of the mechanisms that would be hit by those drugs.
Without getting into the specifics, as we said in the talk, we're prioritising small molecule generics, so already on market with good safety and tolerability data. A lot of them are focused around some of the central mechanisms that we highlighted today. So inflammation, metabolism, that central insulin receptor signaling pathway. So it's very much the mechanisms that we are showing and we have published on. It's just the specific candidates themselves that we're still working on, and obviously trying to reproduce the results in the Decode ME, all of us and other validation studies before we release those results.
Great, thank you. And we have somebody who says they're an Aussie here and they'd like to know which local research group you're running the drug trials for ME with.
We're hoping it's going to be Melbourne, Chris Armstrong's group in Melbourne.
Fantastic, thank you, Steve. Question here about how can you involve my GP to be more aware of this fantastic research. We'll be sharing links. You can access the webinars, and you can also go to the Learn About ME podcasts, which are available through the Action for ME website. So Helen, I'd like to come to you with a question, please. What has been your experience of patient and public involvement, PPI, on the LOCOME project? And has there been anything you've learned or any challenges from doing PPI on a genetics project?
Thank you, Sonia. It's been a really interesting project to be part of, but some of that very detailed genetics, you really do need a PhD in genetics to fully understand. But PrecisionLife, they've been really good at explaining things in a way that is more accessible for us. And I think the PPI partners have also been able to bring their lived experience to aid them. So it's been symbiotic. And we've been able to identify what needs explaining in a more detailed way to the community as well.
Great, thank you. Audrey, I'm going to come to a couple of questions for you. First of all, how long will your study take before results, the one that you've moved on to do, and what is proteomics?
Yes, thank you. And sorry for using jargon. It's always easy to forget what words you take for granted. Proteomics is the study of all of the proteins present in a particular sample. So in our case, we're looking at all of the proteins and how abundant they are in plasma samples. We're hoping to run the experiments this summer. So hopefully we'll have some results sometime next year. I can't promise on what the timeline will be exactly.
Great, thank you, Audrey. We've got a question around drug testing in the UK and how can people sign up. Just to say, we're not at that point at the moment. Part of the, a real drive for us within the ME Genetics Centre of Excellence is about moving from genetics to treatments. And there are a number of other projects around whole genome sequencing, the Sequence ME study, work that PrecisionLife are doing, etc. We are trying to move as quickly as possible to clinical trials, but we do have a way to go. Our recent event at Windsor Castle that was held by Action for ME in partnership with St George's House on the Castle Estate was very much looking at how can we move from the science to trials to treatments, and we are working as quickly as possible. We're just not at that point at the moment. Another question for PrecisionLife. What similarities and differences have you found between ME and long COVID genes, pathways, mechanisms and subgroups?
Christina, sounds like one for you.
Yeah, so well, to start with, as Steve mentioned, we have all of the references to our publications with ME/CFS and long COVID we can provide later, which gives a lot more detail than the quick summary that I'm about to say. We found some similarities and some differences. That comparison is still ongoing, especially with access to the Decode ME dataset now. However, when we look at the UK Biobank ME/CFS population and compare with the long COVID, some of the differences we see in long COVID, particularly those that have more of the all-round severe form of the disease that we hypothesize might indicate a more severe form of the acute infection, we're much more likely to have genes and pathways linked to the SARS-CoV-2 host response and or severe COVID-19. That was distinct from ME/CFS. However, some of the genes and the mechanisms that were found in those with long COVID that had much more of the PEM, post-exertional malaise, classic kind of ME/CFS-like phenotype, we did see similarities around the mitochondrial functionally related genes, cellular respiration, as well as clear signals around the insulin receptor, type 2 diabetes and energy metabolism pathways that we have found in ME/CFS and in the actively protective biology that we saw in the ME/CFS analysis as well, that we didn't quite get to present, but we'll also send the link to the paper for.
Yeah, and that clock gene also shows up in both, and we're seeing not just those individual genes, but genes up and down the pathways, which give supporting evidence that those pathways are likely to be in common rather than just individual genes.
Thank you. We've had a few questions around the funding application that was submitted by the Genetics Centre of Excellence. That's an MRC strategic partnership grant for a project called Prime. There will be a communication coming in the next 2 or 3 weeks, and so we can update you in due course. I've also had a question around Action for ME's commitment to research spend and research activity. That information is covered through our annual general meeting and annual report, so I'm not going to answer that one there, but you can go and read the annual report on through our website. I've got another question for Helen. How have you found being involved in a public-private partnership project?
It's been interesting. I mean, I know PrecisionLife had only done a small amount of ME/CFS long COVID research before LOCOME, so they don't necessarily have the background that some of the other researchers, perhaps people like Chris Ponting, the names we know, have. But they've had a real appetite to learn about ME. They invited me to do a fireside chat for them last year, and it wasn't just the geneticists on LOCOME, it was all their employees came, and that was really positive, and they showed a real interest in what was happening and what's happening in the world of ME and the background. And when the PPI partners have asked for reasonable adjustments, they've been made, and they've done their best to accommodate what that people needed from the perspective of having ME and being involved in the project.
Right, thank you, Helen. And I can also say from Action for ME perspective, it's been fantastic working with PrecisionLife. There has been a lot of learning for all of us, and I think there's been a real openness to growing and developing the patient public involvement work, and that's really positive. And as a genetic centre of excellence, that's something that is absolutely at the heart of what we're doing, including on the partnership board. Helen is one of several other individuals that is working with us on that board to ensure that patient public involvement, lived experience is at the heart. I've got one last question for you, Audrey. Does your study mean that the Ron Davis seahorse study is invalidated, or is that different?
So I think you're referring to the Ron Davis nanoneedle study where they took PBMCs from people with ME and then put them into either plasma from people with ME or healthy controls and measured something called the cellular impedance, and they saw quite stark differences there between patients and controls. It's hard to really know how to compare our results to Ron Davis's because it's quite a different study system. You know, they're using a different cell type and a different measurement. But what I would say is there is a replication study of that happening now. I believe I forgot who is forming it. It's Dr. Fatima Labid and Professor Michael Hughes in Surrey. And they are precisely performing a replication. So we should have an answer as to whether that finding holds true, hopefully soon.
Right, thank you, Audrey. And this will be the last question. I'm going to refrain from trying to answer it and hand it over to PrecisionLife team. Is the hope that Decode ME results will corroborate or expand on your results?
I think we're very hopeful that it will expand on our results. I mean, the ME results that we got, honestly, I'm probably telling tales out of school here, but we very nearly discounted them. They were, it was very difficult with such a heterogeneous disease from such a small population out of UK Biobank, where the data were so poorly diagnosed and characterized in the first instance, to be confident that we had generated really useful results with, at least 15 times that amount of data in a much more consistently collected fashion with much deeper phenotypic surveys. This is going to be much more useful to us, and I think we're all looking forward to being able to run those studies and report on them in the very near future.
Great, thank you. And just as a reminder, we did do an update a week or two ago from the Decode ME team saying that we're working incredibly hard still on the analysis, and we will get results to you as soon as possible. We've had a question coming in around Action for ME and other groups publicising the interim results in the media and news. PrecisionLife are brilliant at pushing out and sharing their results, and particularly on social media, we always support that too. And certainly we will be looking at what we can do in terms of sharing the LOCOME results, specifically that Helen, ourselves and Edinburgh University working on alongside PrecisionLife. So the last thing to say is thank you all for joining us. Thank you to those who are watching afterwards via the recording. And of course, a huge thank you to our panelists and those in the Action for ME team that have worked behind the scenes to make this happen. Thank you very much.